All right, welcome back to class, everybody. I'm going to go to screen share, and I'm just going to go real quick to our Brightspace, make sure everybody knows what's going on. I put the grades up for the past IRAT, GRAT. Everybody got 100, should be good to go. We'll take a quick look at the syllabus, make sure that we're on par. This is February 8th today, and we're going to be doing Module 3, Neural Control during exercise. It's the neuromuscular system. We've already talked about the muscles, their structure and function and sliding filament theory. Now we got to start talking a little bit about the nervous part of that neuromuscular system. So today and Wednesday, we'll talk about the nervous system. And then on Wednesday, we have our IRAT and GRAT for Chapter 2. Chapter 2, as a reminder, was bioenergetics and muscle metabolism. And then we're just going to keep going same way throughout the semester and start to introduce the lab stuff, all right? If you scroll down here, you see the exchange documents been added, and also all the lectures for the spring semester so far are up on YouTube, so take a look at those. All right, if nobody has any questions, then what I want to do is minimize all this stuff and get to our lecture. So let's do so. By the way, if you have any questions, feel free to come on mic at any time so that we can clarify things. And hopefully this will be informative, all right? So Chapter 3 begins with an overview. Specifically, it's an overview of the nervous system. And remember, we keep talking about these system dynamics and the word system and all that stuff. And they call it the nervous system. And in that phrase alone, it implies that these nerves are connected to each other and there are things that are emerging from this system of nerves. And what emerges is the action potential. That's where it comes from. It just emerges seemingly out of nowhere. But according to systems theory, that's something that's reasonable. We'll get into the structure and function of the nerves, how a nerve actually propagates an action potential. All right, now, I don't expect you to go into the finite details of this process, but perhaps just knowing that there's an electrical gradient from one side of the membrane of the nucleus of a neuron to the other, and it's that change in electrical gradient that causes an action potential, I think is probably enough. We'll talk about the central nervous system and the peripheral nervous system. And then the sensory motor integration aspect makes stretching fun, all right? When you're stretching, what you're trying to do is you're trying to manipulate the nervous system, the signals that the muscles are sending to the spinal cord, that signal that, oh my God, my muscles stretching too fast and for too long, clench up or loosen up in the case of your Golgi tendon organ. So when you're stretching, what you can do is you can manipulate the neuromuscular system and you can relax the muscle more and get better stretching. So we'll talk about that later. The central nervous system is the brain, the spinal cord, all right? And the peripheral nervous system are the nerves that are in the periphery of the body. And periphery means like towards the outside. So the central nervous system are the brain and spinal cord. Again, they're thinking about the brain and spinal cord as the top of this information chain, and everything kind of like the marionette hands distribute from there and become the peripheral nervous system. Now let's just do a little bit of work right here. So if you look right here, it says the peripheral nervous system is the afferent nerves. Is the afferent nerves here and the efferent nerves. So afferent starts with A, and A is like an arrow going upwards. So that's how I kind of remember the afferent nerves are nerves that are going up from the body to the brain. And maybe I'll just go like, it's not the greatest drawing of a brain, but let's go like this and just say this is a brain and the brain stem. All right, let's just go like that. All right, so the brain, and now here's the body. This is the periphery. So afferent nerves, let's erase this. Afferent nerves are these ones here, where they send information from the body to the brain. Afferent nerves. And then efferent nerves, these ones here, are outgoing. They send information from the brain down to the body. And this is the efferent ones. And I can remember that because it looks like there's a backbone here, and then there's like things that come from the backbone, kind of like there's a backbone here, and there's information that comes from the backbone and goes down to the body. So anyway, afferent nerves are sensory nerves, like pain nerves and chemoreceptors and baroreceptors and proprioreceptors, and they're giving information to the brain. The brain is going to use that information, and then it's going to respond, and it's going to send information back to the body. And what it does is it has a full loop. This is called a closed loop motor programming. All right. Now, one could argue that that's different than hierarchy. A hierarchical motor programming is that the brain simply sends signals down to the periphery, like just kind of like the brain is the hand up the top here of this marionette that sends signals all the way down to this body that is attached to all of these little lines, and that's how the body moves. So this is pretty much hierarchical motor programming. The brain does everything. This is called closed loop motor programming. And then within the same slide, they talk about systems. So it's like, still, I don't mean to keep hammering down on all these terms, but within this one slide, there are competing theories. I believe in the systems theory, right? Because of those emergent properties and blah, 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 blah. But anyway, you have sensory or afferent nerves coming from sensors in your skin and in your muscles and in your body. And you have also outgoing or efferent nervous signals, and you have those that are going to the muscles, which are somatic, and you also have these autonomic, like kind of, it's almost like automatic things that are happening, and those are things that are happening like your breathing patterns and circadian rhythms and your heart rate and your digestive system, and it's usually going to your organs, your visceral organs. And these autonomic nerves are either sympathetic or parasympathetic. So sympathetic, S, and parasympathetic. One of them is fight or flight. So this is your fight or flight. So it's going to either make it so that your heart rate goes up, gets more oxygen to your muscles so that you can either fight or run. This is your fight or flight somatic. And the way that I remember that is that the parasympathetic nervous system makes things kind of like calm down a little bit. So this is rest and digest. All right. And the way I remember that, 1 second, rest and digest. And we'll just talk a little bit about that. Rest and digest. Now, the sympathetic nervous system is going to make it so that you're better able to fight or run. So it's going to be an increase in heart rate, increase in breathing rate, increase in norepinephrine, increase in vasodilation, vasodilation of peripheral vascular, cardiovascular characteristics so that you can promote more blood to the arms and legs. And the parasympathetic, the way that I remember that is the P in the parasympathetic, it has kind of like this downward part right here. And that to me reminds me that the parasympathetic makes things go down. All right, and that's the rest and digest. So it's going to take the muscles, the blood from your muscles and redistribute it to your internal digestive tract. It's going to make it so that your a vasoconstriction of blood vessels in your arms and legs, and everything kind of goes towards like shutting down a little bit. So those are automatic involuntary actions controlled by your efferent autonomic nervous system. All right. If there's any questions, let me know. All right, so let's clear these pictures. Let's go to the next slide. So this is just another graphic representation of it, right? Information's coming up, going down. Information from your peripheral nervous system in the form of afferent nerves. A is going up towards the peripheral, through the peripheral nervous system into the brain. The brain is sending information now. All right, so there's a couple of problems that we want to discuss here. This word information. Now, this is a really big catchphrase here, especially in the work that I did in my doctoral program, right? Means that the central nervous system, the brain and the spinal cord are receiving all of this information, all right? All of the information from the peripheral nervous system, afferent nerves, Golgi tendon organs, muscle spindles, nosoreceptors, all sorts of different receptors. And then the brain is this thing that is understanding this information. And then the brain is doing something with the information, and then it's sending information back to the peripheral nervous system. I mean, that's a very called information processing. All right, the brain is getting information, it's processing the information in the brain, and then it's sending these signals out. The problem is like when the info comes in, some of these nerves are touch receptors, and some of them are stretch receptors, and chemical receptors, and pain receptors, and temperature receptors, and all of that info, you know, like temperature is Fahrenheit, and pressure is in newtons, and chemical gradients are in, I don't know, plus and minus ion situations. And so the brain is receiving information in all of these different formats. Does the brain transfer all of this information into like one format? And then like if I'm exposed to information, it takes a little time for me to understand the information, to interpret the information. Sometimes this information is stored. So does it change all of these things here and then make it into like binary zeros and ones? Like, is this information stored in English? Is it stored in number form? Is it stored in electrical form, chemical form? And then how does the brain take this information that's coming up and then process it all into usable energy, like usable information, and then use that usable information to change it from degrees or newtons to action potentials so that the peripheral nervous system can run its somatic reflexes or responses or movements or whatever. So, I mean, if you kind of pay attention a little bit, then you'll see that now this is called an information processing, I guess, form of motor control. But that's different than systems theory. That's different than hierarchical motor programming. So there's lots of, and then within it, systems, you know, like it's everywhere. So you still have to just kind of think for yourself a little bit. But I guess this is the closed loop motor programming theory, all right, where information comes in, brain does something with the information. And this is why it's still theoretical, because nobody knows how the brain does something with all of this information that's coming up. And like, I've never really thought about all of this stuff. So is the brain doing it without my input? And if that's the case, then does the brain have a brain and the infinite regress? And all of a sudden you have all these, the same old questions keep arising. But that's a little bit more theoretical, I guess, than we really need to go. So let's go back to the next slide. All right, so pretty sure everybody knows kind of like what a neuron is. It kind of looks like, you have like a nucleus here, and you have all these little dendrite, or these little ways for dendrites from other neurons. You have your axon right here, and then at the end of the axon is going to be your so this is your cell body here, body, and then this is going to be your axon. And then these things here are going to be your dendrites. Just go like den. All right, these dendrites are receiving signals from other neurons, and then the cell body is going to have a electrical charge in it. There's going to be potassium ions on the inside of the cell body, and there's going to be sodium ions. It's not exactly like this. I'll just put them right here. Sodium ions on the outside. And that changes, that makes an electrical gradient between the inside of this particular cell and the outside of the cell wall. And then what's going to happen is that electrical gradient, if the difference between the electrical charge on the inside of the cell is altered or changes past a particular threshold, then the action, we'll get into this, but the action potential will travel down the axon. Maybe I'll even go like this. This changes. Along the axon is these myelin sheaths. They're like these sheaths that look like this. And they help make it so that the electrical signal that's going from the dendrites through the cell body down the axon goes to the next dendrites of the next cell body faster. When you have multiple sclerosis, it's these myelin sheaths that start to deteriorate. We'll talk about that. And then you have your the end of the neuron. All right, so whoops. And the end of the neuron is going to have a number of these little postsynaptic branches that could attach to either another neuron, dendrites, or it could go into a muscle. All right. Some neurons can be big, some neurons can be small, some neurons can be connected to other neurons, some connected to muscles, or they can be parts of big or small motor units. So again, let's go to the next slide. We have our cell body, and in the cell body is a nucleus. All right, and then this cell body has these little processes that stick out, and these are called dendrites. And these dendrites are going to receive signals from other neurons, other electrical signals. All right, and these dendrites are going to send the signal towards the cell body. And if the electrical signals that are coming in, if the electrical signals that are coming into the cell body are weak signals, then there's going to be like a general amount of activity in this neuron, but it's not going to propagate an action potential. If there's a good amount of electrical activity coming into these dendrites, then this neuron will propagate an action potential. So it really depends on what these dendrites are receiving. Then the axon here is where the action potential travels down. Actually, the action potential jumps between these myelin sheaths. It takes time for sodium, potassium, sodium, potassium, sodium, potassium, all these ion channels to open and close. And then what these myelin sheaths do is means that the electrical signal doesn't have to travel down the entire length of the axon. The electrical signal can jump over these myelin sheaths, and it decreases the amount of time the action potential takes to get down the axon if these myelin sheaths are healthy. But multiple sclerosis starts to deteriorate these myelin sheaths. So now all of a sudden this propagation of the electrical signal down the axons in the neuromuscular system is now out of whack and mistimed, and that's why you have like these hypokinetic movements and increased tetanus of muscles and things that makes it more difficult for the body to move like a pendulum. Okay, so the electrical signal for communication between the brain and the arms and legs come from the central nervous system, from the nervous system. But I think that, like the word says, nervous system, rather than thinking of the brain doing it or thinking of the brain being this signal sender, maybe what we can do is we can think of the brain as maybe like a pool of electricity. And a pool doesn't do anything, right? It doesn't necessarily, it just, the electricity is housed in the brain somewhere. And the action potential is more of an emergent property of this system functioning. You know, and now you think about the complexity of this nervous system and the complexity of the emergent property of that system. I think that matches. All right, so now these action potentials, this electrical signal, all right, must be generated by something. So it's either your afferent neurons sending information up to the brain, and then the brain says, oh man, now that I've been given this information, I have to work with that information, do something with it, and then send information back down through the efferent nerves so that the body can function with it. But there are some electrical signals that actually don't use the brain, like your reflex signals are all periphery-based movement strategies. In any event, some stimulus, either visual, auditory, peripheral, some stimulus is going to make it so that the nervous system allows for this action potential to emerge. And then that action potential is propagated, boom, down the axon. And then it leaves that particular nerve and then is transmitted to the next nerve cell or to the next muscle cell or something like that, muscle fiber. At rest, all right, let's erase these things here. At rest, there's energy, there's electrical energy in the cell. And it's actually has a polarization. So the inside of the cell is more negative than the outside of the cell. And the cell, they're talking about this cell here, the cell body of the neuron. And it has a different charge on the inside of the cell versus the outside of the cell wall, because on the inside of the cell there's potassium and on the outside of the cell there's sodium. All right, and these just have different electrical charges. And typically what happens is the potassium wants to exit the cell and sodium wants to enter the cell. But you have these potassium ion channels that make it much easier for potassium to leave the cell than it does for sodium to enter the cell. But because there's an uneven separation of these sodium and potassium ions, it leaves the cell polarized, and that makes it so that it has a resting membrane electrical potential of minus 70 millivolts. And if the potential or the electrical potential within that cell stays pretty close to 70 millivolts, then this particular axon or neuron won't fire any signal to the next neuron. But you could imagine there's a neuron here, and then there's another neuron here with its cell body here, and then there's another neuron attached to this one, and then there's another neuron, and then like this makes the brain. So there's neurons attached to neurons attached to neurons. Sometimes like an electrical signal is going to come into this neuron. It's going to come from like one big neuron up here, and that neuron is going to send electricity, boom, and it's going to get to this cell body, and that's going to send information to this one here, and that's going to send information down. Well, sometimes these little potentials here are known as, one second, a graded action potential or a graded potential. And then there are action potentials. A graded potential is like a very, very small signal. So if like you have electrical signal coming from this neuron, going to the next neuron, going to the next neuron, the the potential, the electrical potential of some of those interactions are just not strong enough to make a big action potential. So you can think of like graded potential as just like information coming, going like down the neurological stream here. But then sometimes there's one electrical potential that makes this 70 millivolt electrical potential in the cell change dramatically. And when this negative 70 millivolts changes dramatically, a full action potential can occur. All right, so let's just see where we're at. All right, so it's why, let's get rid of a little bit of this drawing here. Let's just get rid of that and get rid of that. And let's just do a cell body here. Let's just say this is the cell body, whoops. This is the cell body. And inside the cell, there's potassium. And outside the cell, there's sodium. And they're both ions. They both have a charge to them. All right, and then you have this shuttle here. This is like a door. Then you have another door here. This door, sodium wants to go in. This door, potassium wants to go out. This is your sodium potassium ion channels within the cell wall of your cell body here. And potassium wants to go out of the cell and sodium wants to go inside the cell. And right now there's more sodium on the outside of the cell. So if there's more of a positive thing on the outside of the cell, that means there's less positive on the inside of the cell, and therefore the inside of the cell is negative compared to the outside of the cell. So there's a high concentration of sodium outside the cell and just a medium concentration of potassium inside the cell. So the inside, another way of saying is that the outside is more positive than the inside. But we're talking about the inside of the cell, not the outside. So it's with respect to the outside of the cell, the inside is more negative. It's more negative because of the concentration of sodium outside and potassium inside. And right now, at rest, these sodium channels typically will remain closed. So it's much more difficult for sodium to rush in, and it's easier for potassium to rush out. That means that the outside is going to get even more positive. If the potassium is rushing out, the outside of the cell is going to get even more positive. So the inside of the cell is going to get even more negative. All right. Typically at rest, the sodium can't enter the cell because these sodium channels are blocked. All right. So that there is this, I guess, change or this potential to change this electrical potential difference here because of these sodium and potassium ion channels. And then these sodium and potassium ion channels have these little pumps that keep this at 70 millivolts or close to negative 70 millivolts at rest. So there's like this homeostasis mechanism. But again, sodium, and this is a link right there, sodium-potassium system. And what emerges from sodium and potassium is this negative 70 millivolts. So if I said, where did the action potential come from? Where does the electrical signal come from? Does it come from the brain? Most likely it's some type of emergent property of this system here, interacting with all of these neurons and stuff, but another little hint towards systems dynamics there. All right, so now what happens? Let's go. So a depolarization is when depolarized, it's not polarized anymore. And at first it's polarized to negative 70, but a depolarization is when it goes from negative 70 to closer to 0. Why would it go to negative 70? Why would it get more positive? Well, it's probably getting more positive because either Potassium is leaving the cell, but if potassium is leaving the cell, it's going to get more positive outside of the cell and make it actually more negative. But this is going towards zero, which means it's getting more positive inside the cell, which means that at some point the sodium ion channels open and sodium enters the cell. Whoop. So when an electrical signal is occurring, what happens is this sodium ion channel opens up, it floods inside of the cell, and it makes the cell have more positive ions inside of it, which makes it going to be less negative and closer to 0 millivolts. Now that's required for an action potential or any type of impulse to travel down this axon. All right, so that's depolarization. Hyper is a lot of polarization. A lot of polarization means it's going to go from negative 70 to even more negative, even more polarized. That means either sodium is going to leave the cell, but remember, at rest, there's not a lot of sodium in the cell anyway. So it's going to become more negative when positive ions leave the inside, and that's mostly because this potassium ion channels open and potassium leaves the cell. Now, when that's happening, when you have a hyperpolarization, it's much more difficult for a nerve impulse to actually propagate down the axon. All right. And that's going to affect your neuromuscular firing rate. It might actually make it so that that neuron isn't able to be innervated by an action potential 100% of the time. While a neuron is hyperpolarized, it's not going to be able to receive this action potential and send it to the other neurons down the line or send it to the muscle fibers. All right, so this depolarization and hyperpolarization have this kind of like nervous system function. All right, and if these depolarizations and hyperpolarizations are small orders, not very big, then they're just going to kind of make it so that the system can keep sending information towards each other in the form of these graded potentials. All right, it's strong enough for a signal to pass through the axon, and it can excite or inhibit the next neuron, but it's not big enough to be an action potential. This is like kind of information. These ones here are these graded action potentials are like where, I guess, like just cells can propagate this electrical signal to each other, but it's not going to create an action potential, or it's not going to excite any other neurons that are down the chain so that somatic movement can occur. All right, but if that change is big enough, well, let's go into the graded potentials first. So a graded potential is kind of like a localized change. It's like if one neuron is interacting with another neuron and sending a signal from one kind of smaller region of the brain to the other. An action potential is when that signal is going to leave the central nervous system, travel down through the periphery, and result in some type of movement. All right, now these are generated again by incoming signals from other neurons. So it's pretty much just sending information from another part of the brain to a different part of the brain, or sending electrical current from one side of the brain to the other. All right, but if the graded potential or the potential in that membrane is high enough, then the action potential is going to propagate down the axon and be transmitted to another cell.
Can I ask a quick question about these?
Yeah.
So when it comes to like hearing things, the sensory nerves that receive those signals but don't create necessarily like a physical reaction, is that still a graded potential, or is that an action potential?
You know, I think that we have to almost think of things a little bit differently, maybe, because we're talking about like an afferent situation with the auditory signal, and it's obviously like auditory signal is going to come in as a sound wave. Now, I would bet that if that sound wave isn't high enough, doesn't have a high enough threshold, then it's going to make it so that the auditory neurons are, again, just at this graded potential kind of functioning or kind of status, right? And if that sound signal is high enough, then it would probably propagate that signal down the auditory nerve. Probably that's how it's working. Yes. Now, I don't know enough about auditory nerve stimulation and all that stuff to give you 100%, to be totally honest with you, but I would assume. But then you have auditory nerve, you have visual stimulus coming in, you have stimulus from your proprioceptors and kinesthetic and your otoliths in your ears and stuff. So yeah, I would bet that it's all very, very similar in terms of thresholds of stimulus potential, probably.
Anything?
All right, so let's keep going here. So a graded exercise, a graded potential is a change in this negative 70 millivolt polarized status of that cell body, but it's not strong enough to propagate a complete action potential. An action potential is a large, very quick depolarization of that cell body. All right. And that depolarization is going to go very quickly. All right. So at first, the cell body is at minus 70 millivolts. And if the change in that electrical potential isn't high enough, like if the polarization stays anywhere under negative 55 millivolts, then the signal is going to remain a graded potential. Nothing is going to occur in regards to an action potential being propagated down the axon. So you're not going to have an action potential if you have like a slight change in millivolts, or if the change is, from negative 70 to negative 65. All right, so nothing's going to happen with smaller changes in that particular potential. But if the graded potential or the polarized status of the cell passes negative 55 millivolts, then all of a sudden there's going to be a depolarization of that cell so that it goes from negative 70 to 30 millivolts. So anything between negative 70 and negative 55, it's going to remain a graded potential. But if it gets to 55, then that's going to trigger this sodium ion channel opening. And when the sodium ion channel opens, the sodium floods into the cell. It does the rest of the job to go from negative 55 millivolts and then bang, all the way up to 30 millivolts because of the sodium rushing in. Remember, sodium is positive. When it rushes in and these potassium ions are still closed, it's going to create a big change from negative 70 to positive because positive ions are going in the cell. So if this is like electricity is happening, negative 70 to negative 55, nothing's going to happen. But boom, if there's a big enough signal to send the sodium ion channels open and sodium to rush in, then automatically you're going to go not only from 70 to negative 55, but from negative 55 to positive 30. And there's going to be a big, huge positive spike in that electrical potential within that cell. And that's going to be enough to push the action potential out of the cell down the axon of the cell. Then what's going to happen is there's going to be this efflux. Efflux is like sodium is coming in, efflux is going out. So now you have sodium coming in, All right, and then as sodium is starting to rush back out, there's going to be an opening of the potassium ion channels, so it can go from super positive all the way, boom, back down to super negative. And while this repolarization is happening, this neuron won't be able to assume or to be able to propagate another action potential. All right, so this is just a big change in the electrical potential. If the change is small, then an action potential won't occur. There's sodium rushing into the cell, but just not at a great enough concentration to go and do anything but a graded potential. But if that electrical signal is high enough to get that graded potential to change, it's going to open up those sodium ion channels. It's going to allow sodium to rush in the cell, do the rest of the job in terms of getting that electrical potential from negative 70 all the way to positive 30 millivolts. And then that's going to help propagate the action potential. And then in order to get the neuron back to its resting potential to be able to assume another signal, it's going to have to change from positive 30 to negative 70 millivolts. And it does that by shuttling excess potassium outside of the cell body. And then the cell will be ready to propagate another electrical signal. But again, that doesn't happen instantaneously, right? It happens very quick, but it does make it so that that neuron is not able to propagate an action potential for some time after it propagates an action potential the first time. All right. So this is just a drawing of the same thing. All right, let's move this over. So it says here, maybe I can just erase this here. It says here you have a negative 70 membrane potential. If everything stays here or it doesn't pass this threshold, right, this signal is going to just stay as a graded potential. But if the signal is high enough, it gets past 55, boom, depolarization can occur. Let's go like this. Erase this up here, right? Sodium ion channels are going to open. The sodium is going to move into the cell, making it more positive in the inside of the cell. And then what's going to happen is the sodium is going to have to leave the cell, channels close. All right, potassium channels open. Potassium is going to start leaving the cell, make it more and more negative inside the cell until there's like this overshoot here. You can see this negative 70. This, but then there's a repolarization overshoot past negative 70, and then it like kind of levels back out. All right. When it's in this overshoot period right here, it's much more difficult for the neuron to propagate an electrical signal. Okay. All right, as the signal is going from positive 30 back down to negative 70 and getting back to its resting potential, it's called a refractory period. All right, now as this is happening here, the sodium ion channels, actually on the way up rather, the sodium ion channels are already flooded open, so they can't open anymore. So this signal, once it's propagating an action potential, it can't propagate another action potential as it's propagating one action potential because the sodium channels are already open. All right, so this is its refractory period. All right, and in that case, the neuron is not able to respond to another signal that's coming to it. Again, during this repolarization, same thing. All right, same thing over here. As it's repolarizing, these potassium channels are already open and it can't repolarize anymore. So it's this refractory period and this repolarization period that kind of dictate how fast the neurological system can function. It's not instantaneously fast. All right, there are kind of times where the signals are just inhibited because of just these changing of electrical potentials. I don't think it's necessary to keep going on that too much more. All right, so what is this myelin stuff? Myelin is these little protective sheaths around the axon. All right, omega-3 fish oils and all sorts. I'll show you a video about protecting these myelin sheaths, but they're fatty sheaths, protective things around axons. All right, and these fatty sheaths they're called Schwann cells. And then they're not continuous, meaning that they're like these, there are gaps between the myelin. And these gaps right here are called nodes of Ranvier. And it allows for saltatory, for the conduction of those sodium ion channels to propagate faster down the axon. And multiple sclerosis, that I thought was like terminal, and it's not a good thing, that's for sure. But perhaps with nutrition and diet and exercise, we can protect the brain from the deterioration of those myelin sheaths. And on Wednesday, we'll show you a video about that. All right, bigger axon diameter means that the signal is going to send down the axon faster. So your larger motor units are going to have bigger axons and faster signaling. All right, that's pretty much the discussion on axons and neurons and structure and such. So let's erase all these drawings here. Does anybody have any questions about what we went through? All right, if not, then I'm just going to move on to the next slide. All right, now we're going to start talking about at the end of the neuron, when it gets either to another neuron or it gets to the muscle fiber, it's got to make a connection with something else. So there's going to be a gap between neurons. So you have your neuron here, cell body axon, and then your synapses. And then you have the dendrites of, we'll have another cell body here, and you have your dendrites of another neuron here. And then you have your axon, and then you have your synapses here. Now, you could also have a situation that looks like this. 1 second. This is going to be a muscle. All your striations and these things here attached to your muscle fiber. All right, so there's going to be a gap between the neurons. This gap right here, all right, is the action potential has to cross this gap here. So the action potential is coming down here, and the action potential is in the form of electricity. All right, that's the way that the signal is conducted. Remember, we just talked about 70 millivolts of electrical potential change from one side of this tissue to the other, right? So it's going to change from an electrical signal, and when it gets down into this gap right over here, it's going to change from an electrical signal to a chemical signal in this gap right here. Same thing, when it gets to this gap right here, it's going to change from an electrical signal to a chemical signal. Right, and these chemicals that are going to go from one side of the gap to the other are called your neuromuscular transmitters. There's a couple of neuromuscular transmitters that you might have heard of, acetylcholine and dopamine. And then there's other neuromuscular transmitters that are tied to the hormonal system, the endocrine system, like norepinephrine. We'll talk about that in a second. In any event, you have this axon, and then you have this synapse, and then you have dendrites for the next neuron, and the action potential is going to cross this synapse by releasing some of these neuromuscular transmitters. And these neuromuscular transmitters are going to be released by cells in the presynaptic cleft cells, and it's going to be accepted by cells in the postsynaptic cleft. So the synaptic cleft is this kind of gap here, and these presynaptic cleft cells are going to be releasing these neuromuscular transmitters, and the postsynaptic cells are going to have receptors so that they can receive it. And then once the postsynaptic cells receive the neuromuscular transmitters, then what's going to happen is there's going to be another electrical change. So the signal is going to go from this chemical signal, boom, back to an electrical signal. So there are these changes from electrical to chemical, electrical to chemical. Let's show you here. Next slide. So let's erase this here. So if I took this little circle here and I made that circle bigger, you would get what's in this square right here. So let's just clear all of these pictures. So now you have your neuron, right? This is the synaptic cleft right here. All right. These are the pre-synaptic, before the synapse, axon terminal neuron. So within, this is the neuron. This is the end of the neuron. In here are your vesicles that contain your neuromuscular transmitter. In this case, it's going to be acetylcholine. This is the synaptic cleft, and on the opposite side is your postsynaptic, your after the synapse receptors, and they're receptors that are receiving these neuromuscular transmitters, this epinephrine or dopamine or whatever. So you have electrical signal coming down, and now you have a chemical signal, and those receptors are going to connect to, or I guess link up to, or be constrained by these neuromuscular transmitters. And then the neuromuscular transmitter is going to allow for an electrical signal to go into the muscle fibers, allow calcium to be released in troponin and tropomyosin, and then the muscle will contract. As long as the change in potential, electrical potential, caused by these neuromuscular transmitters is high enough. Same thing, like in order for the axon to propagate a signal, there needs to be a high enough membrane potential. Same thing. These neuromuscular transmitters are probably being accepted by these receptors all the time, but just not at an extent that's high enough to cause a contraction. But when this interaction happens high enough and it creates a high enough electrical change within the muscle fiber, then the muscle contraction can propagate. All right, this action potential can't go this way. The action potential comes down only in one direction. All right, and it releases these neurotransmitters, dopamine, acetylcholine, and these are chemical messengers that carry the electrical signal, bind onto the postsynaptic surface, and they stimulate another graded potential in that postsynaptic neuron. Now, it remains a graded potential unless the electrical signal that crosses the synaptic cleft is high enough to create a big enough electrical potential change. And if that's the case, then what happens is there's going to be an action potential rather than just a graded potential in the postsynaptic neuron. I know that some of this is dry. Drew, we good?
Yeah, all good.
Okay. All right, so a neural, this is like, now this right here, it could be talking about going from one neuron to the dendrite of another neuron, all right? And when you're talking about transmitting action potentials from one neuron to another, this is gonna hold true. But now what we're gonna talk about is a specialized kind of junction called a neuromuscular junction, the junction between a neuron and a muscle. All right, acetylcholine is going to propagate this action potential, and it's going to excite that muscle fiber so that you can have the intertwining of actin and myosin. All right, acetylcholine is going to bind to a receptor on the muscle fiber. It's going to depolarize, allow the action potential to go down the plasma lemma, release calcium from the T-tubules, allow troponin and tropomyosin to interact. But one of the limits that we have to human motion is that we do have that repolarization period, right? So that will slow down the propagation of the action potential to some extent. Here's your neuron, it comes down, it interacts with the muscle. We can blow that up and look over here. Same thing that we saw before. You have your synaptic vesicle that has your acetylcholine, your synaptic cleft that goes right into the muscle fiber. Neurotransmitter is going to be accepted by the receptors on the motor end plate, and there's going to be a graded potential unless this propagation of action potential and the receptors are, or the reception of that neuromuscular transmitter is high enough to create a high enough change in potential, and then there'll be a propagation of a of an action potential down the muscle fiber into the sarcoplasmic reticulum and affect the actin-myosin. Now, acetylcholine is one of the main neuromuscular transmitters associated with muscle contraction, but there are many other types of neurotransmitters, many different kinds. And then this neurotransmitter here, norepinephrine, all right, is released by the adrenal cortex. It's also called adrenaline. So adrenaline is a hormone, and that hormone is used to alter sympathetic nervous system effects. So sympathetic, remember, it doesn't have the P. So sympathetic is to increase heart rate, increase. That's the fight or flight one. So now not only do you have acetylcholine, which is a neuromuscular transmitter, but now you also have a hormone. Now, what's cool here is not only do you have neurons, which are sending electrical signals, but now all of a sudden you have hormones. And now not only do you have the neurons working, but now the neurons are now also going to be connected with hormones. And now you almost have like a neural hormone system. And what's going to emerge from neural hormone system is another emergent property in and amongst itself, but that's going to help for and make it so that there's an even greater control of the body's ability to utilize oxygen, carbohydrate, and bioenergetics and fuel. All right. All right, so let's go to the next slide. Drawings. And again, those in the post-synaptic, after the synapse, all right, the response that's going to happen after the synapse is the action potential is going to be generated as long as the change in potential from one side of the membrane to the other side of the membrane is high enough. If it's too low, then the muscle will remain with some type of electrical activity, but the muscle won't contract. So the acetylcholine, the neuromuscular transmitter, crosses the synaptic cleft, gets accepted by the receptors. That propagates an action potential. The action potential runs down the muscle fiber. The action potential is interacts with the sarcoplasmic reticulum. That releases calcium from the T tubules. That's going to allow for troponin to move off of its troponin and tropomyosin to move off of the binding sites, and then actin and myosin can interact with each other. I'm sure that's what this is going to say here. All right, depolarization excites and promotes the action potential, and then so there are some action potentials or some neurotransmitter signals that excite this postsynaptic potential, which means that it depolarizes, makes things less negative. All right, and there are other signals that inhibit the postsynaptic potential, hyperpolarizing, make it so that it's potentially more negative. These inhibitory things are making it more negative, so from -70 to -90, and now it needs to work even harder for an action potential. So these things inhibit the action potential. All right, or you could have excitatory, which is things that are going to go from -70 to +30, which is going to help promote the action potential. So sometimes these neurotransmitters can do different things. And that's pretty much our discussion of neurons. All right. If it's okay with you guys, I would like to take a 1-minute rest. And what we're going to do then is we're going to get into the different parts of the central nervous system, the brain. But give me a second. All right. Let's see here. Get a sip of water. All right. So we've spoken about the neurons, and there's neurons in the brain and neurons in the spinal cord. So at least we have like the basic functioning unit of the central nervous system. And there are people who have, you know, and achieved doctorates on neural anatomy and cognitive sciences and stuff. As I've implied before, my doctorate and what I learned is that it's not the brain controlling everything. The brain is like a functioning part of this system. Again, it's a central nervous system. So it's not the brain that's responsible for motion and movement and memory and thinking and emotions, and those are things that are emerging from the system of parts, which is the cerebrum, diencephalon, cerebellum, brainstem, and all of the afferent nerves that are coming up. Those things are more emergent properties as opposed to the brain being the thing that's controlling them. The brain is also, when you like look at the brain and you look at the gray part of the brain, it's typically you're looking at the cerebrum. And the cerebrum has two different sides to it. It has a left side of the brain and a right side of the brain. And the left side of the brain and the right side of the brain are connected. All right, connected by the corpus callosum. And let's just talk about the sides of the brain for a second. The left side of the brain, boom, boom. And the right side of the brain, boom, boom. Now, the left side of the brain is different than the right side of the brain. They do different things. The left side of the brain is responsible for things like math and reading and kind of like fine-tuned details. It's typically somebody who has a good like vocabulary, or my dad was very left brain. He could come up with all of these different little facts and things, and he was really good reader and could remember you know, just little details about things. So the left side of the brain is more, I guess, like it's more informational, it's more I guess to the point. It's more verbal. We can even say verbal. The right side of the brain is a little bit different. The right side of the brain is more associated with things like music, imagination. Let's see, music, imagination. If you're lost in the woods and you got to get yourself out of the woods, it's the right side of the brain that's going to get you out of there. It's awareness. It's creativity. I'm not saying that people who are left brain are only, you know, verbal and not creative, but I have a business partner, and I have the ability to come up with these ideas and these dreams and stuff, and I'm very creative, but sometimes like I don't know how to use Excel like really effectively. But my business partner is a whiz at Excel, but like he can't talk to people. Like he can't call people in customer service and really do anything effective like that. He gets kind of, he's too verbal. I mean, he's too kind of like rigid. He's too structured. But I can talk to people. right? I come up with the ideas. He does all the grunt work behind the ideas. He's very left brain. I can't even tell a joke. He'll be like, you know, it takes everything literally, right? I play music. I write. That's what I do. I kind of like do things creatively. So I'm very right brained. He's very left brained. And because of that, we kind of don't get in each other's way too much. We complement each other. Typically, an individual who has Parkinson's disease, they have a problem with the basal ganglia. We'll talk about that in a couple minutes. It's a part way inside the brain, and what Parkinson's usually starts on one side of the brain versus the other. You could think of like the basal ganglia, kind of like maybe look like this, right in the middle of the brain. It's not what it looks like. I'm just drawing. And when the basal ganglia deteriorates, all right, all the functioning of the basal ganglia also deteriorates. And inside the basal ganglia is, we talked about where dopamine or levodopa, dopamine is generated. And remember, dopamine is a neuromuscular transmitter that is working in opposition to your acetylcholine. So dopamine makes your acetylcholine makes your muscles want to contract and propagate that electrical potential, and dopamine is going to kind of counter that. So an individual who has Parkinson's disease has a deterioration of this basal ganglia, but usually it happens only on one side versus the other. Sorry, more appropriate. Usually it starts on one side and then it kind of propagates itself through the basal ganglia eventually. So sometimes, depending upon the symptoms of the individual's Parkinson's disease, you can determine whether or not they have LPD, left Parkinson's disease, or they have right Parkinson's disease, right side Parkinson's disease, right? And individuals who have left and right Parkinson's disease, if they do affect one side of the brain first versus the other, then individuals who have Parkinson's disease, sometimes they'll have a hard time recalling facts that they used to know, or speaking, or doing detail-oriented things. And if that's the case, it's maybe a hypothetical that they have Parkinson's disease that's affecting the left side of their brain before the right side. Sometimes individuals have a hard time walking, or sometimes they veer or bump into things, and it could be the case that those individuals have Parkinson's disease that's affected the right side of their brain prior to. And then a good amount of my doctoral research was separating these two groups of people, putting them in a virtual reality environment and seeing whether or not the LPD subjects veered or changed their walking strategies more or less than the RPD subjects. And if right side Parkinson's disease is associated with spatial orientation, then maybe these right-sided Parkinson's disease subjects had greater veering when they were put into these environments, these virtual environments. And sure enough, that's what we found. So that's pretty much the basis of my doctoral work, was looking at the effects of a deterioration of the basal ganglia and how that affects movement and how that affects this whole central nervous system, right? Right. So if a part of this cerebrum or diencephalon or cerebellum or brainstem, if a part of this system changes, then what emerges or what happens is different movement strategies. So if the part of the diencephalon, which is the basal ganglia, if that starts to deteriorate, it could be the case that the system changes. And when the system changes, what emerges also changes. So in any event, let's go back to here and I can erase some of these pictures. All right. The brain, as you know it, is a cerebrum, is two cerebrums, a left and right, that's connected by a corpus callosum. So now these two spheres, these two hemispheres can communicate, or these two hemispheres are connected to each other, or there's a cerebrum system, right? Two cerebrums connected as a system. All right, there's the cerebral cortex, which is the outer layer of the cerebrum. All right, and it's typically the gray matter that, you know, if you look at a brain in a picture, it'll be gray. That's what you're looking at, is the cerebral cortex. And then sometimes people will refer to that as the conscious mind or the conscious brain. But then, right now you have completely, totally new discussions of what is a mind, what is consciousness, what is awareness, where does it come from, right? But let's just go on the hierarchical motor programming theory, the conscious brain or the mind. Like, right, this is all theoretical right now. It says the word mind. We can't prove where it is. We can't show where it is. We don't even know where the mind is. So now we're all into theory. But the outer layer of your brain is the cerebral cortex. The cerebrum has 5 different lobes. If you're looking at a brain, the brain will look like, I guess, somewhat like this. There's like another lobe in the back here. There's your brain stem that comes out here. This is your frontal lobe. This is your parietal lobe here. This is your occipital lobe back here. In the middle here is your temporal lobe. Sorry, whoops, that should be a T. If you fall down and you bang the back of your head, or even if you take your hand and you kind of like bang on the back of your head just a little bit, kind of like I'm doing right now, all your visual areas will kind of get all messed up. Or like if you've ever like put a massager on your neck, and like you look at the TV and it's all like wavy and everything, it's because your occipital lobe is being impacted by that massager, so that it's changing your visual input, and you interpret the signal from the TV differently. The frontal lobe is associated with motor control. Now, this is a very mechanistic view of the brain. Like, mechanistic meaning what you do is you take like a car, or you take like any type of machine, and you can take the parts of the machine apart and say, this is what this part of the machine does. That's what this part of the machine does. So you're like sectioning off the brain, you know, and saying, here's one part of the machine, here's another part of the machine, here's another part of the machine. This part of the machine does this. This part of the machine does this. This part. And that's kind of like, you know, the body isn't a machine. The body is like necessarily, it's a system, and the system works together. So to necessarily say that, you know, all of the motor control is and comes from the frontal lobe, all right, that's pretty much, you know, another example of hierarchical motor programming, saying that this motor control here is a function of the frontal lobe. And then all auditory interpretation is from the temporal lobe. You know, what you're doing is you're assigning now the frontal lobe that particular obligation. And I don't know, it's just something to think about. Okay. All right, let's move on. All right, so the frontal lobe is the primary motor cortex. It's where conscious control of skeletal muscle movement occurs. I don't know if you've ever like practiced imagery, mental imagery, positive affirmation, but there's even this this golf CD that I listened to. It's called The Subconscious Mind. And what you do is you practice over and over and over and over and over. And your practice is working on conscious control of skeletal movement. You set up and you do 100 putts in a row, and you try to make sure that your club head is this way and your feet are that way, and you put the ball in your stance, and you keep your wrists locked, and you keep your grip the same way. And every time I go back to the golf course and I work on that stuff, I'm working on the conscious control of my skeletal muscle. And I do that so many times that what happens is the unconscious kind of mind takes over. So when I get to a golf match or a tournament, I'm not thinking of the face angle of my club or where my feet are. Now I can let my subconscious mind take control of my movement. In any event, what we would say is that the conscious control of skeletal movement comes from your frontal lobe, from the primary motor cortex. It starts with action potentials and then travels down a series of related neurological parts that eventually terminate in the spinal cord, and that those action potentials will crisscross through the spinal cord and go down to the peripheral nerves and eventually to the muscles. Again, we talked about other regions in the brain that are interesting, especially regarding Parkinson's disease, is the basal ganglia. They help initiate sustained or repetitive movements. Like a repetitive movement is like walking. When I walk, my legs go forward, backward, forward, backward, forward, backward, forward, backward, forward, backward, forward, backward. And they get this like in phase or out of phase rather pattern with each other. That is, walking is similar to running in the sense that you have this repetitive thing. Chewing is another one where your mandible just goes up and down and up and down and up and down. And usually you chew for 30 seconds, this sustained or movement pattern that needs to be, that needs to occur for a long time. The basal ganglia has a lot to do with like, it's like an inner timer, like an inner like metronome of the body. And when the metronome is off, then the orchestra can't play together, and there's less of that time constraint. So things start to get a little bit hyperkinetic, a little bit difficult time, especially among individuals who have Parkinson's disease, with like walking like a pendulum and letting their arms and their legs swing with gravity. So typically what happens is it increases the amount of muscle tone. And not only that, but because the basal ganglia creates dopamine, and dopamine is the thing that reduces the excitation of the action potential in the postsynaptic cleft. If there's too much acetylcholine and not enough dopamine, that's going to increase the amount of muscle tone. All right, so let's move on here. The diencephalon is where the thalamus and the hypothalamus are. And what the thalamus is you can kind of think about the, and the hypothalamus is like kind of almost the heat dial, like, you know, in the nest, or like you have the thermostat. You have your thermostat, and the thermostat is going to regulate the body temperature and regulate all sorts of other things that need to kind of be at homeostasis, like appetite, fluid, the amount of sleep that you have. But remember, the hypothalamus is now a gland, all right? It's an endocrine gland where there's going to be a release of hormones. So one interesting thing about the hypothalamus is that it's like, now you see neuroendocrine, neurological, and hormone. So the neuroendocrine system is what we're going to talk about next week. And now not only do you have this neurological system and you have a hormonal system, but you have a neuroendocrine system here, right? And now all of a sudden the neuromuscular system is going to be linked to the hormonal system, and then the emergent properties of linking two major systems together, or the neuromuscular system, the digestive system, the cardiovascular system, the endocrine system, they're all linked together. And the emergent property of all of those links, that's what life is. That's how we're able to kind of function. So I don't think it's the brain doing it. I think it's the emergent property of all these systems functioning together. But in the diencephalon, or the inside, the inner brain, is where the hypothalamus maintains the functioning environment inside the body. It maintains osmolarity, temperature, heart rate, blood pressure, sleep cycles, eating cycles, digestive system, and all that stuff. The cerebellum is that little thing in the back of the brain. So if you have the brain kind of looks like, I guess, like a terrible drawing is like this, right? And then in the back, you have that kind of like the lump here, and then you have your brainstem down there. So this is the cerebellum, and it's in charge of more complex movements. Now, timing is important. Timing and sequencing. Now, you could have 100 people in an orchestra. 100 people. They're your best musicians. Everybody has gone to like Berkeley or some other type of Juilliard or some other school of playing music. And they all get to their very first practice of the evening, and you're going to play Mozart Sonata in C, and you give everybody, they've never played it before, you give 100 of the best musicians this one musical piece, and you tell them to play it for the very first time all together. And most likely, even though they're the best musicians, most likely, if they don't have, especially if they don't have a conductor, most likely everybody's timing is going to be off. When the violins come in, and when the timpani comes in, and when the oboes come in, and how loud the brass section should play compared to the drums or whatever, the timing is going to be off, or the sequencing is going to be off when the violin comes in compared to the bass, drum. I don't know. And if the timing and sequence of the firing of these electrical signals is not effective or hasn't had the time to emerge, then these movements, or then this musical piece, is going to sound muddy. What the cerebellum does is it allows for the establishment of timing to be developed. And what it does is it actually uses your proprioception, or your afferent nerves, to tell the brain what is going on, what's going on, where are my limbs, how fast are they moving, what am I doing. It sends information from your periphery all the way back up to the brain. And according to this theory, the brain figures out what it is doing now and compares that with what the brain intends to be going on right now. And then it sends a correction in the form of efferent nerves back down to the limbs so that movement can happen. Now, again, how this happens, how this comparing, this is where the whole theory here breaks down. Because what you're doing is you're saying that you have this black box, and it's a black box theory in the sense that this black box is called the brain. And what happens is information comes into this black box like this. In the black box, stuff happens, and then just like Charlie in the Chocolate Factory, this correction spits out. Now, as long as we keep this a black box and say it's the brain, if Drew is like, but Dr. Young, how does that happen? How does the brain compare anything? When does the brain compare? Where does the brain compare? Does the brain compare, does it say, I wonder what I should be doing compared to what I'm doing now? I've never actually been privy to that response in my brain. I've never woken up and did any of that stuff. It seems as though it just happens. So the reason why they call the brain a black box is because once this afferent information comes into the brain, we can't really explain what happens in here to make this signal come out of the brain. And I don't think we'll ever be able to. So thinking of the brain as this comparer of information that consciously initiates everything is difficult. And that's the thing that accounts for body position and muscle status. Right? Before, we're talking about, systems. And now all of a sudden, this phrase goes back to hierarchical motor programming theory, where the brain is doing all of this stuff to account for body position. It receives input and helps execute and refine movements. We're saying the cerebellum does that. So does the cerebellum have like decision-making like capabilities? And if so, does the cerebellum have like a small brain inside the cerebellum? And now again, you're getting into your infinite regress and all of these questions. So at some point, you have to be a little bit skeptical at some point. Not all skeptical, but a little bit skeptical. Or not skeptical. I mean, you're going to have to be willing as you go into your graduate programs to think about like what is really occurring here. All right, so let's go brain, cerebellum. Let's go to the next slide. Let's see where we're at. All right, the brainstem. Obviously, the brainstem is going to be relaying information from your spinal cord to your brain and your cerebellum. All right, let's get rid of this. Right, there are different parts of the midbrain, different parts of the brainstem, the midbrain, the pons, medulla oblongata. All right, the reticular information, the reticular formation is where skeletal muscle function and tone is coordinated, along with your cardiovascular system. I don't think that this is all 100% necessary for you to know all of this neural anatomy. All right, so just be aware that there are different parts of the brain. People try to attribute each part to different responsibilities, which is a mechanistic approach. And I think that we could probably move on to the spinal cord. All right, so the spinal cord has afferent and efferent nerves. This is going towards the brain. This is going away from the brain. All right, sometimes the brain is involved with movement. Sometimes the brain is not involved with movement, like reflexes, like pulling your hand off a hot stove. All of that, that movement comes or is initiated by excitation at the spinal cord. But information can go up towards the brain from afferent or sensory fibers, like your Golgi tendon organs or your muscle spindles. We'll talk about that in a little bit. All right, your peripheral nervous system is are the nerves of your arms and legs and your organs, and there are a number of different connections to the brain. And for the most part, these cranial nerves, spinal nerves are going to distribute electrical signals and action potentials to skeletal muscles. Right, there's the sensory nerves and the motor nerves. Sensory nerves are like chemoreceptors, baroreceptors, pain receptors that are sending information from your skin or from your sweat glands or from your body so that your brain can function or can parcel that information so that it can send out effective motor action potentials through the efferent nerves and allow for motion to occur. So you have different types of receptors in your periphery, different types of receptors in your tissues. You have thermoreceptors that are responsive to temperature. Now, temperature is going to be degrees. Mechanoreceptors are responsive to things probably like newtons of forces. Nosoreceptors are responsive to pain. So light, chemicals, these are all in different information. Physical forces are usually newtons. This is temperature. This is chemicals. This is photons. Right? All of these things eventually are going to go up the afferent nerves and get to the brain. And according to the black box theory of the brain is going to be able to take all of this different kind of information, transfer it to this brain information, calculate all of that information, and send the information back to the muscles through efferent nerves. All right. If that's the case, say for instance, I have photoreceptors and I'm receiving light. All right, it comes in as photons. And then at some point, I have these photons, and these photons are going to have to, photons. Wow, it's having a hard time drawing. Hold on, let me erase that. Yeah, for some reason this is choppy. Give me 1 second. Let's just go photons. So at some point, photons are going to interact with the rods and cones on the back of your eye surface. And those photons are going to go to your brain. And then the brain is going to have to do something to those photons, the black box again. And then inside the black box, it's the brain. There's going to be some type of calculation here. And then the brain is going to send information back to the body so that the body can use that information. Now, one of the things that we want to consider here is these photons. If I open my eyes, all right, I get information into my eyes. Some people will say that, like, if you're looking at a tree, when it finally gets into your eyes, remember it gets into your eyes, and then you have the upside down tree on your retinas, and your brain has to figure out the upside down tree. So the point is when I open my eyes and I look, the photons that come into my eyes are not information enough to be able to use. The information has to be processed by the brain. And only after it's processed by the brain can that information be used by the body. Okay, this is called indirect perception. Indirect. It's called indirect perception. And say, for instance, I'm using my ears. All right, well, the same kind of would hold true. The sound, the audio signal, all right, the audio signal that gets into my ears, oh boy, audio signal. The audio signal that gets into my ears, okay, is not good enough. It has to get into my ears and then go into the brain. And then the brain calculates or does something with the sound, and then it spits it back out like Charlie in the Chocolate Factory so that my body can function with it. Or I have these nociceptors, or I have temperature, and the temperature again goes into the brain and it does something with it. This is all called indirect perception, and it's indirect because I don't perceive these photons or the sound or the temperature. What I do is I receive that information, but only after the brain does something can I actually perceive it. I receive information, the brain does something, and then I can perceive the information. So this perception, right? This perception that occurs, or the ability to use that information, is indirect. It's indirect because it takes an extra step. And the extra step is the brain. The brain does it. But there's also another competing theory of perception called direct perception. Direct perception. All right. And that was developed by a guy called James J. Gibson. All right, back in the 50s. James J. Gibson says that right now, this photons and stuff, it's interacting like, here's a photon, here's a brain, and when they're connected, they make a photon brain system, and what emerges from that photon brain system is this, perception. So it's not a black box thing. There's no reason for the brain to have to do this step, this indirect step of understanding what all of these receptors actually mean. Gibson is saying that these signals are the connection. They are this thing. And when light interacts with my connector, which is my eyes and my optical nerve, all right, this just is the line that connects photons to the brain or to that electrical potential. And what emerges from here is perception. So with James J. Gibson, he is saying that this whole black box step is not necessary. The perception of you when I look at you happens without this black box. All right. Typically, when you see stuff like this, mechanical receptors, physical forces, goes in, transmits information. Is this in Spanish? Is this information in photons? Is it in audio? How does the brain translate all this information? Well, if it's indirect perception, meaning the brain does all of this calculation, then you have to answer this information problem. And not only that, but like, let's say photons come in. The photons are of this tree down here. And I look at the tree, all right? And the photons end up coming into my retina. They hit my rods and cones. They're upside down. That information is in photons, all right? How long after this information hits my brain, how long after it gets into my brain do I have a perception of it? Like, let's say here's a timeline, all right? Timeline. Right here is at the beginning when the photons are not in my brain or don't connect with my eyes. And now it's right as soon as after the black box, after the brain has done stuff. So right here is the beginning of the timeline and the end of the timeline. The beginning of the timeline is right here, photons. The end of the timeline is right here, when the brain is about ready to push its perception onto the body. How long into this process did the perception happen? Did perception happen over here? Did my brain have to, perceive at least half of this upside down tree before it could start to understand what it is that it was perceiving? If information is the thing that the brain is taking in and the brain is calculating the information, like for me, in order to understand or in order to like take in the information of a lecture, it takes me like a week of studying to be able to do what I'm doing, So how long after the brain sees these photons does the brain decide, oh, that's a tree? Because if you're thinking that the brain is taking in information, the way that information works is that information isn't perceived instantaneously. It's indirectly perceived sometime later. So if we're indirectly perceiving things sometime later, after this brain does all the work, then wouldn't we be delayed? Everything we did would be delayed by some amount of time, and I don't feel like I'm delayed at all. I feel like this information comes in, or these photons come in, and this perception, like I showed you the other day, when you just open your eyes and you can perceive a whole landscape in one visual like attempt, it seems as though these photons and the brain interact so that perception emerges directly. The information that comes in is not impoverished. In indirect perception, information is called impoverished. It's not informative enough as it is here for the brain to understand. It's impoverished. It needs to be understood, or it needs to be changed. The information coming in needs to be calculated or something in order for the information to be used. Indirect perception is based on impoverished information, but direct perception is not. James J. Gibson said the information in the environment, the photons, the audio in the environment is informative enough so that the brain doesn't have to calculate anything. Therefore, the information is directly perceived, direct perception as opposed to indirect perception. I believe after doing all my studying in direct perception and not indirect perception any longer. All right, does anybody have any questions about that? I know that's a little bit dense, but right, and it's not just photons that are coming in. It's sound, audio, light, pain, temperature, forces, newtons, chemicals. All right, and not only is it those things, but it's all of these things as well. Muscle spindles, Golgi tendon organs, joint kinesthetics. All of these are informative signals in and amongst themselves that don't need to be calculated by the brain, right? They're just different ways of connecting the brain, photons, audio, and what emerges from all of those links and all of those connections is, boom, perception. All right, and then you don't need to have processing time. It doesn't take time down the number line for perception to occur. It happens directly. If you change the photons, if it's a Tatiana versus a Cassie, then the photons are different. The way the light reflects off of both of you is different, and what is perceived directly is a different person. I'm never making a calculation of which one I'm looking at. All right, but in any event, some of this stuff gets a little bit dense. So, but I think it's stuff like this that I think about that makes it so that I can have an imagination now about movement and about all this stuff. And instead of just learning something, like I hope that you're not necessarily learning anything, but what you're doing is being exposed, because when you learn it, it means it's static. It means it's done. And I don't believe in learning like that. I believe learning is being exposed to things so you keep asking questions and thinking about things. All right. So you have other sensors as well. You might think of just your eyes and your ears, or even temperature or chemical gradients, but there's other things that allow us for sensory information to be sent back to the brain. You have things in your joints. So if you close your eyes and you lift your arms and you make a letter T with your arms, even though you're not looking at your arms, you can tell that your arms are horizontal to each other. All right, because you have kinesthetic receptors in your joints. These kinesthetic receptors are sensitive to the lengths of your muscle and tendons. So I'm sure you've probably heard of muscle spindles and Golgi tendon organs. And if you haven't heard of muscle spindles, you've probably been in the doctor's office where they do the little hammer on your patellar tendon, and that makes your leg bounce. Well, what they're doing is they're messing with your muscle spindles. What a muscle spindle is, I think we get into that in a little bit. Hold on. Let's see. All right, so so those are your sensory nerves. We'll get into muscle spindles and Golgi tendon organs later, because I believe a good stretching regimen or a good bout of stretching is not to necessarily elongate the muscle, but it's to mess with the muscle spindles and Golgi tendon organs so you can get a little bit of a greater range of motion. So we'll talk about that in a bit. So this is the sensory receptors. All right, all the information up neurons. All right, but then you also have your motor neurons. Motor neurons are going to send information from the brain down to either your muscles or your organs. All right, autonomic is your organs and somatic is your voluntary skeletal muscle. But then we also maybe want to talk a little bit about voluntary. What is voluntary versus involuntary, or what is a reflex versus not reflex? So I'm going to ask you guys a little bit of a question. 1 second. Voluntary. What is voluntary and what is a reflex? Maybe I'll ask somebody to come on mic so I can ask you a few questions. All right, so what is a voluntary contraction versus a reflexual contraction? Let's talk to Aubrey. Why not? How are you today, Aubrey?
I'm good. How are you?
I'm doing pretty good. All right, so let's just talk, you and I, for a little bit about a voluntary contraction or voluntary movement. All right. Aubrey, can you tell me what you think is a voluntary movement?
Voluntary is when you think about it happening.
Okay, I agree with you totally. All right, so voluntary is when you think of emotion occurring. So, and maybe you can tell me what you think is a reflex.
A reflex would be something where like it happens when you don't think about it. Kind of like those like the pain stimulus.
So let's just say think and don't think. I think that's probably pretty easy for people to remember. And I would tend to agree with you, at least from what I thought, okay, what I was taught a long time ago. All right, but maybe this is a little bit different. So, Aubrey, could you give me an example of maybe I'll give you an example and you can tell me whether or not you think it's voluntary or not, okay? Now, Aubrey, I don't know if you have a cup of coffee there, but if I told you, Aubrey, I want you to go grab a cup of coffee, grab the cup of coffee. Do you think that that's voluntary or a reflexive action?
Voluntary.
Right, it's pre-thought of. I initiated the behavior with some type of stimulus. Right, I'm giving her an auditory stimulus, and so I would also agree that is a voluntary movement. All right, that you thought about. Now, can you give me a reflexive movement that you think is something that I wouldn't have thought about?
Something that you wouldn't have thought about as a reflexive.
Reflex. What's a reflex? Any example of a reflex?
Like if you're like, say you have like a mosquito bite and then it's like itches and then you like go to itch it. I don't know. It's like a reflex. It's not like you.
I would say.
I mean, that could be
I would say
too.
Reflex is like if somebody throws a ball at you, it's about to hit you in the eye, and what you're gonna
And then
you're gonna have that movement strategy to try to like preserve your health so you don't get injured. All right, or like pulling your hand off a stove. All right. I don't know if anybody's done any work with babies, but there's a reflex, a bunch of different reflexes that you do with babies once they're born. There's the swimming reflex where you can put the hands and feet in the water and the baby will swim. Or there's the writing reflex, or there's if you lift one hand up, the other hand goes down, or the other leg goes up. But there's another one called the Babinski Test. Does anybody know what a Babinski Test is?
Isn't that the one on your foot?
Yeah,
where
exactly.
It grows or not?
Yep. So tell me what you think of what you know about
it. Isn't a positive Babinski is when like the doctor will drag like their finger or like a pen like along your foot and your toes like curl, and that's supposed to happen like as a baby, but as you grow older you kind of grow out of it and then you don't have that reflex anymore.
Exactly right. Okay, exactly right. So let's just maybe redefine it. Okay, a Babinski test is, especially when they do it with a little baby, newborn, just like Aubrey says, they'll take the foot and they'll take like a little a pen or like a little, I don't know, something, one of those tongue depressors, and they'll scrape the bottom of the foot, and initially the baby is its toes are gonna retract. Okay, then they take so that's a reflex. Everybody would agree with that's a reflex. It's not thinking. The baby's doing it without thinking. All right, but when you keep scraping the bottom of the foot over and over, you do it once, the baby's foot retracts. You do it again, the baby's foot retracts. You do it again, the baby's foot retracts. But if you keep doing it over and over, what's going to happen, Aubrey?
Like, does it become like a learned behavior?
Yeah, just as Aubrey's saying, if you keep doing it over and over to the baby, eventually the baby's going to realize that this isn't something that I have to retract from anymore. And it's going to stop doing that kind of retraction with its toes eventually. So here you have this reflex, this Babinski reflex, which is, as Aubrey said, it's a non-cognitive, no thinking necessary movement. But if I do the reflex over and over and over again, Aubrey also said that I'm going to learn that I don't need to do that reflex anymore. So here we thought that the reflex was not cognitive, but now I can unlearn a reflex. So this line of what's a reflex and what's not a reflex, and can a reflex be unlearned? Because if it can be unlearned, I can probably learn it. And now here's another thing you might want to think about. Aubrey, let's say you are doing a voluntary contraction and you're going to go reach for your coffee cup, and your coffee cup is over on the other side of your desk. All right. Even though you're thinking about grabbing the coffee cup, and that's going to be like a cognitive action, what's the first muscle that fires when you're going to go reach for that coffee cup?
My arm.
I mean, you would think it's your arm, but as soon as you lift your arm up and your arm goes out, it's going to create torque, and that's going to make your body want to tilt towards your raised arm. But when I lift my arm, my body doesn't tilt. And it doesn't tilt because before I even lifted my arm, my opposite side lower back muscles contract isometrically to stabilize my body so I don't tilt over because of the change in center of gravity. But I never really thought of my lower back and core muscles firing so that I could stabilize this voluntary contraction. Right? It seems as though as a result of this voluntary contraction, there was a reflexive contraction that happened in my lower back opposite side. So now all of a sudden, a reflex can arise from a cognitive action. I didn't think that that was the case. And you can also learn and unlearn a reflex. And now all of a sudden, my definition of voluntary versus reflexive is also now in question. And I'm not 100% sure what the difference between the two is. Right, and all of a sudden, these little things that my professors would point out to me, I'm like, man, it seems as though everything's kind of like getting thrown into a washing machine of information, and it's hard for me to now understand exactly what I'm thinking about. But those are the things that allowed me to kind of like free myself from having to learn what other people were teaching me and start to kind of think of things for myself.
So voluntary movements just involve reflexes then?
I mean, I think that I don't know if I can necessarily fully define each of them, Cassie. I think that what you're going to have to do now is understand that, you know, there are probably vocabulary that you've been exposed to that now needs for you to kind of think a little bit further. And what do you think? Is a voluntary contraction or a voluntary movement completely voluntary? And if that's the case, then what are your definitions? If the brain doesn't do everything, and voluntary could be involuntary and reflex could be thought of, and the eye's not like a camera, and like, what is it now at the end of all of this? What is it that you're going to believe? What is it? You know, and I think that that's going to take a lot of maybe introspection from each of you. It's going to take a lot more reading, a lot more work, and it's going to take a lot of, at some point, it's going to take for you to, like, you know, jump all the way in the pool, you know, find a theory of motor control, or find a theory of, you know, personal training or physical therapy, or find a theory of sports psychology that works for you, and then you're going to have to kind of go in all the way and be able to just use that as your theory. Well, I'm not sure, right? But I know it's not what I used to think it was. So you have this somatic nervous system, which is, stimulating voluntary skeletal muscle activity, but who's to say what voluntary is anymore? Not 100% sure. All right, and you also have this autonomic nervous system. The autonomic nervous system is where you have like not muscle activity, not voluntary muscle activity, but the activity of your lungs and your heart and things that seem to kind of go automatically. And you have two divisions of your autonomic nervous system, your sympathetic and parasympathetic nervous system. And again, we talked that your sympathetic nervous system is your fight or flight nervous system. When you want to fight, you want to get a lot of blood flow to your muscles. Your heart rate wants to go up. You want to get the blood out of your digestive tract into your muscles so that you can have oxygen for, a bout of fighting or a bout of running. So all the things that are going to make it so that you have more energy, there's going to be an increase in glycolysis. There's going to be an increase in lipolysis. There's going to be increase in vasodilation of your peripheral, vascular system. There's going to be a restriction in the blood vessels in your gastrointestinal tract. All stuff to make it so that you have fuel, oxygen, and sugar in your muscles so that you can get out of there. And that's going to be excited by your adrenaline. That's why they call it adrenaline, because it comes from your adrenal glands, and it increases heart rate and increases vasodilation, help get oxygen to your body. Your parasympathetic nervous system is the opposite. The part of the P that goes down reminds me that everything drops in your parasympathetic nervous system. So a reduction in heart rate, a redistribution of your blood towards your intestines so you can digest and go to sleep and kind of relax. So you have your sympathetic and parasympathetic nervous system. Your sympathetic nervous system, like I said, is fight or flight, increase heart rate, increase blood pressure, increase blood flow to your muscles, increase airway diameter so you can get oxygen, increase the ability to burn in bioenergetics and burn fuel, and increases mental activity. I gotta either fight or get out of where I'm at. And you can feel that all happen. It happens relatively quickly when you get into a situation where you either have to fight or flight. And again, your parasympathetic nervous system is the rest and digest. All right. It's the opposite of your sympathetic nervous system. Increases digestion, conserves energy, reduces heart rate, reduces the diameter of your blood vessels and airways, saves oxygen, saves carbohydrates, saves fuel. All right. Stores energy for the next bout of sympathetic nervous system activity. And this shows an example of changes to your heart muscle, your blood vessels, your lungs, your liver, with sympathetic and parasympathetic effects. So heart muscle, you would think that your heart is going to have to get more oxygen to the body if it's trying to fight or run away. So your heart increases its heart rate and also the force of the contraction during sympathetic nervous system excitation. Your coronary blood vessels, they are the vessels that are going away from your, towards your heart, towards your lungs, and that's going to cause vasodilation so you can get more oxygen into your heart tissue. More oxygen to your heart tissue means a more effectively functioning heart. Your lungs are going to go through bronchodilation. Broncho is lungs, or the tubes that bring air to your lungs, and dilation means get bigger. So there's going to be easier for you to pull oxygen into your body. Your blood vessels, there's going to be an increase in blood pressure, mostly because there's an increase in your force of contraction. All right, there's going to be vasoconstriction. Vaso is blood vessel, constriction is getting narrower of abdominal viscera. That's blood vessels in your stomach, blood vessels in your gut, so that it's going to try to get the blood out of your gut and back to your skin. Vasodilation is get bigger. There's going to be an increase in the size of the blood vessels in your skeletal muscles and in your heart, so that you can have more oxygen into those tissues. The liver is going to stimulate glucose release or glycogenolysis. All right, cellular metabolism goes up, meaning that there's going to be more energy transferred for fuel. Adipose tissue, it stimulate lipolysis. Lipolysis is fat. Lysis is breaking apart of fat. All right, so it's preparing your body for a reduction in glucose, because glucose is fuel. So in this fight or flight thing, the sympathetic nervous system, your body thinks that there's going to be spending carbohydrates soon. So we might need extra carbohydrates. So it's going to, in your liver, make the glycogen in your liver go through glycogenolysis, the lysing of glycogen, to make glucose. Sweat glands are going to increase their sweating rate so that you could prepare for increases in thermodynamics and temperature. Adrenal glands, your adrenal glands are right above your kidneys. The adrenal cortex is where epinephrine and norepinephrine, which is adrenaline, is secreted. And when we talk next week about hormonal control, like if you take a drop of ink and drop it in a bucket of water, it takes a little time for that ink to diffuse throughout the water, similar to how long it takes for norepinephrine to get into your bloodstream and get to your muscles. It's not immediate. That's why one of the things you want to do is go through a good warm-up, a 5 to 10 minute warm-up. That will help increase all of these sympathetic nervous system functions, and it'll also give enough time for the norepinephrine, which was secreted during the warm-up, to get into your bloodstream and to help with all of the rest of this stuff here. During sympathetic nervous system activity, you want to try to get all the blood to your muscles and get it out of your digestive tract so that you can have fuel and oxygen for contraction. So typically your digestive system will kind of shut down a little bit, which is why I don't want my guys, eating too much during workout. If they want to have some carbohydrate, they can do that, but I don't necessarily want them to eat, Sarku Japan during the middle of workout, because it's just going to make them heavier, because there's a decrease in the digestive system functioning. So I don't want that. And then there's a decrease in urine formation so that your body can maintain its water. And remember, when you break down ATP, there needs to be hydrolysis in order to do that. And if you don't have enough water because you're secreting too much fluid in your urine, then it's going to be more difficult to break down ATP and have enough water for hydrolysis to occur. All right. Does anybody have any other questions about that stuff? All right. Apparently your sensory and motor systems are integrated. I mean, this is another systems approach. All right. Between the sensory and motor systems, there's an interaction. There's a constraint between the two. All right. Stimulus is going to be sensed by your chemoreceptors, by your eyes, by your ears. It's going to trigger an action potential. All right. That action potential is going to be sent down your sensory neurons all the way up, afferent neurons, to your central nervous system. The black box is going to start. The central nervous system somehow is going to interpret all of this information. Don't know how it does that. This is all theoretical. You won't be able to answer that. And then it's going to send out a response. All right. This is the whole reason why this is all theoretical. Once it sends out this response in some form or another, one second, let me just change this. Once it sends out a response, there's going to be an action potential sent down the motor neurons, and those motor neurons will arrive at the skeletal muscle, and then the movement will occur, and that's going to result in potentially another stimulus, and it just keeps going on and on and on and on. The loop is closed. This is called closed loop motor programming theory. Closed loop motor programming. All right, where you have signals from your muscles or from your skin, and that's sending information. There's a nail in the skin, right? That sensory neuron is sending an action potential to the central nervous system. The central nervous system black boxes everything, right? This central nervous system interprets the information and determines the motor response. How does it do that? When does it do that? How fast does it do that? How can it do it for a little baby who's doing something for the very, very, very first time? How can the central nervous system interpret the information if you have somebody who's like on a soccer field running at you and their information is always changing? Or is the central nervous system able to understand instantaneous change? And how does that happen? One second, let me get back to that. So how does the central nervous system interpret this information? What form is the information in? How long does it take for the information to be perceived? All of this stuff is in question, right? And then the action potential goes to the muscles, creates some type of movement, and that movement is going to create some other type of sensation or whatever that continues the loop. Here's an animation for the same slide. All right, and then when we get to our, I think that maybe we can stop here. I think that maybe we can do that. I only have a couple of slides left. I want to talk about muscle spindles and Golgi tendon organs on Wednesday, so we have something to talk about. We can talk a little bit about stretching and how Golgi tendon organs and the information sent back to the brain can make a muscle tighter or less tight. And then I think that's probably a good place to start. So I'm going to go like this and stop my share. Does anybody have any questions about anything? I know it's 10:15, but we just went for a good, what was that? When did we, it was 2 hours and 15 minutes on that info. I think that's probably pretty reasonable. Does anybody have any questions? 1, 2, 3, 4, 5, 6, 7, 8. Make sure that you're ready for your IRAT GRAT on Wednesday. If you have any questions or anything, feel free to text me. Text is so much faster. Send me an email on the de.young@assumption.edu, but hit me up with a text too, because it goes right to me and I'll be able to help you. All right?
I have a question about the IRAT.
Yeah.
Are we gonna be able to use our notes for this one, or was that only for last week?
I mean, I don't really mind. There's nothing I can really do to control that. So I think that, and as it was last week, even using your notes, not everybody got 100%. So I think that I want to make it as doable for you as possible. So why don't we just do, maybe we can do, instead of a half an hour, we can do 20 minutes. It seemed as though we had plenty of time to get it done in 20 minutes. So why don't we just keep going with what it was? I don't mind if you use your notes, and we'll do the best we can. Okay? Anybody have anything else? All right, hope you have a nice day.